Module 1, Part 6. In Part 6 of Module 1, we are going to talk about isolation of cell organelles or subcellular components. These techniques are very important and constitute some of the core and routine techniques to study cell biology. The first step in isolating organelles from cells is to disrupt the plasma membrane. In the case of plant cells, you have to disrupt the plant cell wall. This disruption must be very gentle in order to preserve the structure and function of the organelles. So, disruption is usually achieved with the presence of an osmoprotector. Often, the cells are disrupted in the presence of 0.25 molar sucrose solution, which protects the integrity of the organelles when the cells are disrupted. There are a number of ways to disrupt the plasma membrane, such as the ultrasonic vibration, sonication, tissue homogenizer, high-speed blender. You can also force the cell through a filter with small pores, usually 8-micron pores. It disrupts the plasma membrane without damaging the organelles. So once you disrupt the cells, you have a homogenate, sometimes called an extract. The homogenate contains a mixed population of particles of different density, shape, charge, and size. Because of the unique feature of all these particles within the homogenate, you can take advantage of this and use centrifugation to further purify the organelles because the organelles have different density, shape, charge, and size. They have different sedimentation rate. This is why you can apply centrifugation as a second step. Here in this process, often PMSF is used as a protease inhibitor that we learned in the part 1 of the module. The most common second step in organelle purification is differential velocity centrifugation. This diagram outlines the principle of this type of centrifugation. First, you filter your extract or homogenate to remove clumps of cells or unbroken cells. Take the filtered homogenate and apply to a low-speed spin for a short period of time. So in this diagram, you would centrifuge the filtered homogenate at 600 G for about 10 minutes. The resulting pellet contains cell nuclei. So if you only collect the pellet, you can study the nuclei from the sample. But if you want to study other cell organelles, you can take the supernatant from that pellet and apply higher speed spins. In this example, it is 15,000 G for 5 minutes, and under those conditions, you will pellet mitochondria, chloroplast from plant cells, lysosome, and peroxisome. You can take the supernatant and subject it to higher spins or longer spin. In this case, 100,000 G for 60 minutes. You will pellet portions of plasma membrane and the endoplasmic reticulum and some of the larger polyribosomes that might be free in the cytosol attached to the endoplasmic reticulum. Further higher speed spins will give you small polyribosomes, and eventually you will end up with a supernatant that is representative of the cytosol. Now you have purified the organelles, but may not be pure enough. So let's focus on this pellet that contains mitochondria, chloroplast, lysosome, and peroxisomes and see if it can be purified even further. All of these organelles have different densities, and you can take advantage of another type of centrifugation known as equilibrium density gradient centrifugation. The characteristic feature of this type of centrifugation is that the centrifugation is taking place through a density gradient from a low density material at the top and a higher density material at the bottom. One of the common material is sucrose. Sucrose density gradient centrifugation is a standard method of purification. There are some commercially available solutions that can serve as gradients as well. The purification scheme is that you take your organelle fraction of interest and you layer it on the top of the gradient, and you apply your centrifugal force, and the organelles will move through the gradient, and when they reach an area of the gradient that is equivalent to their own density, they will stop moving. So after centrifugation, you will end up with bands of purified organelles. So in this example, the lysosome will end up at 1.12 grams per ml, mitochondria will end up at 1.12 grams per ml, and peroxisomes will end up at 1.23 grams per ml. So how do you isolate these purified organelles from the gradient? You start by collecting the fractions or layers of the bottom of the gradient. So to collect the highest density to lowest, you collect set volumes into different tubes. For example, you collect 100 microliters in one tube, another 100 microliters in a second tube, and another, and so on. Once you identify which tube has your organelle of interest, then you can conduct the biochemical or functional studies on your purified organelle. So let's say we are interested in determining all the proteins found in the mitochondria that you have purified as shown in the previous slides. We want to define what is known as mitochondrial proteome, every protein in those mitochondria. So you can do this by mass spectrometry. This is a diagram that shows the principle of mass spec. So how it works is that you have a sample that you are trying to characterize, and the sample is absorbed onto a metal target. Pulses of light from a laser will ionize the proteins and peptides in the mixture. They become charged. There is an electric field in the analyzer that accelerates the particle towards a detector. All particles will move at different rates. The time that takes for the particle to move from their original position to the detector is known as the time of flight, which is proportional to the square root of the mass of the particle over charge. Or basically, the lightest ion will reach the detector first. The molecular weight of each ions from the sample is calculated based on the time of flight. So you purify the mitochondria as mentioned, and the scheme is shown on the left. The purified mitochondria is then digested by a protease like trypsin, which cleaves the carboxyl side of lysine and arginine, and will do so for every protein in the mitochondrial sample. This sample now contains numerous peptides mixture from the trypsin digestion. This sample is then absorbed onto a metal plate. You ionize the peptides, they move in the electric field towards the detector, and each peptide will have a characteristic time of flight. What you do is to compare the time of flight of all proteins in the sample with the known time of flight of proteins with a database, and by doing so, you can identify almost every protein in your sample. This is a very powerful approach to characterize the proteins in the organelles of interest. There are many other techniques to characterize other macromolecules in the cell, such as DNA and RNA, but we will not be considering these in this course. This concludes part 6 of the module 1 and also concludes module 1.